How do cell cycle checkpoints act as your body's ultimate defense against cancer? Because the disease fundamentally begins when just one cell loses its internal regulation and divides too fast. So let's track a single newly formed cell as it begins its lifespan. Before it can multiply, it must complete a strict sequential growth pathway called interphase. During this track, the cell copies its genetic code, which is a highly vulnerable process, because replicating DNA frequently introduces errors. To stop a damaged cell from passing on harmful mutations, it halts at built-in inspection barriers. At the G2 checkpoint, regulatory proteins act like a physical stop sign, verifying the DNA for damage. If the inspection fails and the DNA is broken beyond repair, the proteins force the cell to produce self-destructive enzymes. This programmed cellular suicide is called apoptosis. But what happens when the genes making those regulatory proteins mutate? The inspection barrier fails, and the stop sign disappears entirely. Without that check, the damaged cell divides continuously, cloning itself into a dense, uncontrollable mass of rogue tissue. And that single starting cell from the beginning of the track, without its breaks, it just transformed into the seed of a malignant runaway growth.